Interphase is the phase in preparation for mitosis. It consists of G1 phase, where the cell grows, S phase, where the DNA is replicated, and G2 phase, where the cell prepares to divide. Prophase is the first stage of mitosis. In prophase, the chromatin fibers become more tightly bound, condensing into discrete chromosomes. The nucleoli disappear, and each duplicated chromosome appears as sister chromatids. The mitotic spindle begins to form, and the centrosomes move further away. In prometaphase, the nuclear envelope begins to fragment. The chromosomes have become even more condensed, and each of the two chromatids of each chromosome now has a kinetochore, a specialized protein structure located at the centromere. Non-kinetochore microtubules interact with those from opposite pole of the spindle. In metaphase, the centrosomes are now at the opposite ends of the cell. An imaginary plate begins to form where the centrosomes lie. The kinetochores of sister chromatids are attached at the kinetochore. Anaphase is the shortest stage of mitosis. Cohesion proteins are cleaved and two sides of the chromatid pair. The cell elongates at the kinetochore, and the two cells have equivalent collections of chromosomes. In telophase, two daughter nuclei form in the cell, and the nuclear envelope develops. The chromosomes become less condensed. In cytokinesis, the division of the cytoplasm occurs, and the cleavage furrow pinches the cells in two. In prophase 1 of meiosis 1, chromosomes begin to condense and homologs loosely pair along their lengths, aligned gene by gene. Crossing over is completed while homologs in synapses are held tightly together by proteins by their lengths. The synapsis ends mid-prophase and the chromosomes in each pair move slightly apart. Each pair has one or more chiasmata, and the centrosome movement, spindle terminator, and nuclear envelope break down. In metaphase 1, pairs of homologous chromosomes are now arranged on the metaphase plate. Both chromatids of one homolog are attached to the kinetochore. In anaphase 1, the breakdown of proteins responsible for sister chromatid cohesion along chromatid arms allows homologs to separate. Homologs move towards opposite poles guided by spindle. Sister chromatid cohesion persists at the centromere, causing chromatids to move as a unit. In telophase 1 and cytokinesis, two haploid cells form. Each chromosome can still consist of two sister chromatids. At the beginning of telophase 1, each hap has a complete haploid set of replicated chromosomes. Each chromosome is composed of two sister chromatids. One or both chromatids include regions of non-sister chromatid DNA. Cytokinesis usually occurs with telophase 1, and two haploid daughter cells are formed. In prophase 2 of meiosis 2, a spindle apparatus forms. In late prophase 2, chromosomes, each still composed of two chromatids associated at their centromere, move toward the metaphase 2 plate. In metaphase 2, chromosomes are positioned at the metaphase plate. Because of crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical. The kinetochores of sister chromatids are attached to the microtubules extending from opposite poles. In anaphase 2, the breakdown of proteins holding the sister chromatids together at the centromere allows the chromatids to separate. The chromatids move towards opposite poles as individual chromosomes. In telophase 2 and cytokinesis, the nuclei forms and the chromosomes begin condensing and cytokinesis occurs. Meiotic division of one parent cell produces four daughter cells, each with a haploid set of unreplicated chromosomes. Each of the four daughter cells is genetically distinct from the other daughter cells and from the parent cell. Meiosis 2 produces four haploid gametes.